Hi everyone, my name is Emery. I'm part of the NIH Oxford Cambridge Scholars Program, which means I'm doing a PhD but split between here and the National Institutes of Health in the US. So I've been in Oxford for about the past 2 years, and I'm going to go back to my lab at the NIH soon. And a lot of what I'm talking about today has been developed in the NIH lab called the Surgery Branch in the National Cancer Institute. By a man named Steve Rosenberg since the 1980s, and he's still working there today. So none of this would be possible without him, and so that's a lot of the backbone on what I'll tell you today. So I'm overall going to tell you about turning your own cells into cancer therapy. So we're first going to start with a little bit of background immunology on how this is even possible. So the cells we're talking about are T cells, and in this very specific interaction, they're able to recognize cancer cells. And this immunologic principle has been utilized to make a treatment with T cells, primarily in melanoma or skin cancer. And I'm then going to talk about the transition to using this therapy for metastatic solid tumors. So these are cancers of the pancreas, your breast cancer, colorectal cancer, so the largest killers in cancer today. And these are cancers that are metastatic, so they've spread all throughout the body, so they're the hardest to treat and have the worst prognosis. But this therapy that's been pioneered in skin cancer is now translating to some of these even harder to treat cancers. And so overall, I'll kind of give you an overview of this general new fourth principle of cancer treatment called immunotherapy. And as I go throughout the talk, you can kind of be thinking about how this type of cellular immunotherapy maybe compares to other cancer treatments you may be more familiar with. And so kind of the hint here is your classic cut, poison, and burn. So this is surgery, chemotherapy, and radiation, how we've treated cancer for the past decades, and how this new way might be a little bit different. So the backbone of this process are T cells. The job of T cells are to find invaders to protect your own body. So these are a white blood cell that circulates all around your body looking to tell self versus non-self. So how do they make this difference to, kill foreign invaders while protecting your self cells? Well, every cell in your body has to tell who it is. It has to prove itself. And they do this by taking their internal contents, their proteins, chopping them up, and presenting them on the surface. And they don't just put them on the surface, they're carried in this little molecular basket called the human leukocyte antigen, or HLA. So every cell in your body has to show its internal contents, chopped up, called antigens, on this HLA. And this whole complex of antigen plus HLA is recognized by a T cell with its T cell receptor. And this is a very specific and precise interaction, such that when the antigen, the little chopped up protein, is your own protein, it comes from self, the T cells don't kill. But how about in a non-self situation? So say our cell is infected by a virus, and instead of your own proteins that get chopped up and put on the surface, it's a viral protein that gets put up there. So we have a viral antigen. Well, now the T cell receptor recognizes this complex, says that's not self, and is able to kill the infected cell. So there's a huge amount of diversity involved in this process, where each T cell in your body has its own T cell receptor, and you literally have billions. Like, your body takes your DNA, shuffles it up randomly to have trillions of combinations of possible T cell receptors, and that's in each of our bodies. We all have different T cell receptors, so there's so much diversity. And the point of this is to make sure that any pathogen out there at a population level where someone is able to recognize and defend against it. So a very diverse situation. But how does this relate to cancer? Well, cancer comes from self, but it has DNA mutations that transform these cells to be different than self. So this DNA mutation that makes cancer can be turned into a protein that gets chopped up and presented on the surface. But it no longer looks like a self protein anymore. It's not a normal self antigen. It's now a neoantigen. It looks different. And so the cancer is kind of telling on itself by presenting on its surface the very thing that has made it cancer. Now a T cell can come around and kill that cancer cell. So this process of a cell getting mutated, transforming, but being recognized by a cancer cell and getting killed happens all the time in your bodies throughout your life. But eventually, the cancer cells are able to override this process, they win, and you have a tumor in the first place. But you don't just have a tumor with cancer cells. There are also T cells in there. that have failed to do their job, but they do reside in the tumor. So back in the 1980s, researchers thought, okay, well, maybe if the T cells are in there, they're there for a reason. They were seeing the cancer, but they just weren't, maybe there's not enough of them anymore. They haven't done their job. So we just need to give them a boost. So they had a patient with metastatic melanoma, so skin cancer that spread all throughout the body. They took the tumor out and they grew the T cells in that tumor out. So these are tumor infiltrating T cells. T cells are also called lymphocytes, so tumor infiltrating lymphocyte therapy or TIL therapy. So they grew out the T cells that had been in the tumor, thousands and thousands, 10,000-fold, up to billions of T cells that had been in the tumor. And this made up the treatment product. So they reinfused these billions of cells to the patient. And so this was tried for a long time in metastatic melanoma skin cancer patients that had progressed through every other therapy. They've had chemotherapy, they've had surgery, they've had radiation. Nothing has worked. They've progressed through everything. So about 10 years ago, before the advent of some other immunotherapies, metastatic melanoma, if you presented with that disease, you had about a 6-month median survival. And in 5 years, about 10 to 15% of patients would be alive. So it was a very, very devastating disease. So how about these patients for whom nothing else has worked? Well, TIL therapy in about 30 to 50% of patients with metastatic melanoma works. They have a response, their tumors shrink, and this is successful. And if you see this red line, this very long tail, this is a survival curve, so we're just looking over time at patients who are surviving. About 20% of responses are complete long-term. So these are people who nothing else worked, they would not survive, but 20% are functionally cured. So these are 10 to 20-year responses where the T cells from their own tumor, grown up and given back, were able to cure those patients for whom there was no other option. So this is really exciting, and it's impressive that for a long time, people thought the immune system had nothing to do with cancer. And now we've proven cells from your very own body are able to cure tumors that are very aggressive and don't respond to anything else. So in 2024, this led to the FDA approving this treatment called lifileucel for unresectable or metastatic melanoma. And so this is exciting, and now patients in the US and Europe actively get this therapy for their tumors. And it's being advanced and tried in more types of cancers and earlier on to improve its efficacy. So let's do a little overview of TIL therapy for melanoma. It's good because of how personalized it is. It's very, very specific to your own tumor because it's made up of cells recognizing your own tumor. And T cells are incredibly potent. They can kill every last cancer cell in your body. That is their job, to protect you from foreign invaders, and cancer is a foreign invader. But they're still constrained by these natural immune safety mechanisms. They don't kill self cells, so it's a very specific but potent treatment. And so because of this, when you have a complete response, it's very highly durable. You get one infusion of these billions of T cells, and if it works, you are good to go. No other treatments needed, because these T cells stick around and continue to protect your body, whereas other treatments only work when you're getting the treatment. But it's not all great. There are difficulties. One of which is the high cost. So this is an incredibly tricky therapy to make. It requires very clean conditions and a tumor to be resected, these T cells to be grown up, and it takes time and specific infrastructure to have this done. And so this is a big limitation to being able to do this at a larger scale. But as more and more therapies of these types, so cellular therapies, develop, this infrastructure is getting better and better, and these therapies become cheaper. There are also toxicities during treatment, and so this mostly comes from some of the drugs that are given alongside the T cells to help them work better. And while these are reversible toxicities, it is still not an easy experience to get this treatment. And while it's been really exciting in melanoma, skin cancer, it hasn't worked nearly as well for other solid tumors. Your pancreatic cancer, your breast cancer, your colorectal cancer, that make up an even larger burden of cancer mortality. And so why is this? So in melanoma, we painted this really nice picture where there's a tumor, there's T cells in the tumor, and they're there for a reason because they were recognizing the tumor. All we needed was to give a boost. In reality, a lot of the T cells there are just on their normal circulation route passing through the body, and they just happen to be in the tumor when it gets resected. They're just bystanders. They recognize other stuff, maybe viral things, maybe bacterial things, but they're not recognizing the tumor. They're just there. So even when you expand the tumor infiltrating T cells, a lot of them are just nobodies. Another reason is the harsher environment in solid tumors that makes it harder for T cells to do their job. So T cells have these natural off-ramps where they don't kill as much over time. And this is good because you don't want T cells going crazy all the time in your body. A lot of what makes you sick, what you feel bad when you're sick, is your immune response, which is a good thing, but you want these off-ramps so that doesn't happen forever. But in cancer, when T cells start to take these off-ramps, even when they haven't done their job of killing the target cell, killing the cancer cell, this is not good. So you have a T cell in this harsh environment that is specific to the tumor, but these off-ramps have been activated and now it's exhausted or dysfunctional and it's not killing anymore. And so you overall have more exhausted, no longer functional tumor-infiltrating lymphocytes. So overall, even when we expand these TIL, the tumor-infiltrating T cells, 10,000-fold to a billion cells, you still don't have enough functional tumor-specific T cells to actually get the job done. So what do we do about that? Well, we can get even more specific. So we can actually find the T cells and their T cell receptors that are doing the job of recognizing the cancer. They recognize these neoantigens made up of the cancer mutations. So how do we do this? We first have to compile all of a patient's tumor mutations. So all of the things the T cells might be targeting, the potential neoantigens derived from these tumor mutations. And then we take another type of cells from a patient's blood that's able to present antigens. And so now we have a platform which to present all of the potential neoantigens, the mutations. And we add these neoantigens made up of the mutations onto these antigen-presenting cells and combine it with the patient's tumor-infiltrating T cells. So from this combination, we're able to find which exact T cells and their T cell receptors are doing the job of recognizing the cancer neoantigens. And so this is exciting. We now have a bank of known neoantigen T cell receptors. So we know they're specific to a tumor. And so now if a new patient walks in and they have that same mutation and the molecular basket, the HLA combination, we already have a T cell receptor we know works for their tumor. So instead of utilizing the cells in their tumor that might not be specific enough or might be dysfunctional, we can just take their healthy T cells in their blood that might recognize all different things, and we engineer our known neoantigen cancer-specific T cell receptor in. So now we've taken a bunch of random T cells and made them cancer-specific T cells. And so we can infuse that back to the patient. So as an entire treatment product, all specific to a given cancer mutation. And so this is very exciting because, again, it utilizes those same mechanisms of potent T cells, specificity, and the safety mechanisms that come along with that specificity. But you can imagine some of the difficulties in it's limited to neoantigen-specific T cell receptors we already know, and a patient matching that exact combination, which is a rare occurrence. So despite those limitations, this is currently being done in clinical trials in the US and other labs around the world to improve this therapy for patients that have no other options left for their cancer treatment. And so overall, I've kind of given you this image of T cells being a very powerful way to treat cancer. And so where do I come in? While it all is very cool, it doesn't work all the time, as we saw in the melanoma case, and as we see even more so in solid epithelial cancers. Despite all of the specificity in our T cells, it's not always quite enough. So the more we can learn about the neoantigen-specific T cell receptors and how they're seeing the tumors will allow us to pick the right T cell receptors to do the job. And so this is obviously a very nice cartoon we have here, but the actual interaction is quite complex. And so I study exactly how loads of different neoantigen T cell receptors do their job of seeing cancer cells and how we pick the right ones to make better treatments. So back to back to our question. Given this kind of new type of therapy, we can think about how does this differ from our classic surgery, chemotherapy, and radiation, and what are some pros and cons of that? Thank you.